over the past decade or so, we've been seeing a tremendous multi-institutional effort around the world to sequence and characterize diffuse large B-cell lymphoma. In this EHA, we saw a culmination of a multi-institutional project, quite fantastic, of over 400 patients with primary mediastinal B-cell lymphoma. Primary mediastinal is a very intriguing lymphoma because it sits on the spectrum between Hodgkin's and diffuse large B-cell lymphoma. More in young patients, we see certain characteristics of Hodgkin's, certain characteristics of diffuse large, and therefore it's quite homogeneous in terms of its molecular pathways. So there is it's addicted to the JAK-STAT pathway. We see a lot of amplification of on chromosome 9 in the location of the PDL1, PDL2 genes. We see recurrent mutations in SOX1s, which we also see in Hodgkin's lymphoma. You may see them in nodular lymphocyte predominant Hodgkin's lymphoma. So a very interesting molecular or biomolecular entity, and a huge endeavor when you understand that genomics. The most common mutations can sometimes be maybe 30% or 40% of the patients. You understand that you really need multi-institutional collaborations in order to elucidate knowledge and to identify small subgroups. And this group effort identified basically mutations in CD58 to carry poor prognosis. And we know that this gene is associated with immune evasion, and mutations in DASP2, which is also works on the MAP kinase pathways, like IRAK4, to be associated with a better response. And they saw a lot of epigenetic changes, particularly in one of the zinc fingers, that explain some of the genomic instability and the genomic changes that we are seeing in this very interesting disease.